Welcome to this fascinating and highly relevant webinar that is titled Probiotics as Invisible Allies to Promote Our Health and Prevent Diseases. My name is Caroline Linninge, and I'm working as a Senior Research Manager at BioGaia. I have a background in microbiology and nutrition, and I will be the moderator of this webinar. And in the webinar, we will have great opportunity to know more about the human-microbe interactions. And we will also have the opportunity to hear about the scientifically proven evidence related to probiotics and the oral cavity, and the concept of oral to systemic. You will know more about BioGaia and how we work to increase the knowledge of probiotics. And finally, you will have some information about the foundation PAR, that works to prevent antibiotic resistance. And our prominent guests for today is Gianfranco Grompone, and he is the Chief Scientific Officer at BioGaia, and he has a background in microbiology. We also have Jacob Axelsson, and he has a background in immunology, and he works as a research manager at BioGaia, and he is in charge both of clinical trials and to develop the preclinical research. And Christine Koppelhus, she is the Director of Scientific and Public Affairs at BioGaia, and she has a background in microbiology, food science, and nutrition. And after the presentations, we will have a Q&A where you can put your questions to the speakers. So let's start with a presentation of Gianfranco. Please, the screen is yours.
Thank you, Caroline, for this introduction. Welcome, everybody. I'm really glad and honored to participate to this webinar. Today, we're going to discuss about the fascinating world of the microbes, the good microbes which are living with us, and their role in preventing diseases. So let me start with this quite provocative concept. If we think about us, we are not necessarily humans. We are literally microbial containers, microbial ecosystems. Within us, in our bodies, we have trillions and trillions of microorganisms living in specific places. In our skin, in our guts and intestines, in our mouth, in our lungs, in our vaginas, etc. So all the places in our body which is in contact with the external world are colonized by these microorganisms. And most of them are bacteria, but not only. And we're going to talk a little bit more about how these microorganisms can help us in our day-to-day life. Today, these observations led scientists to propose a new concept in the evolution. And we are not talking anymore about Homo sapiens. We are literally holobionts, which means we are symbiosis ecosystems where humans and microbes co-evolve together in what is called today the hologenome theory of evolution. And why hologenome? Because we have genes, we carry the genes of these microbes in more than 150 times more than our own genes. So this led also scientists to identify what are the microbes which are living with us. So I mentioned the bacteria. These bacteria are extremely important and are probably the most known microorganisms living with us. If you take the example of the colon, like in this slide, you could see that we have not only bacteria, but we have also virus. The virome, intestinal virome, is extremely important, and it's a field of research which is very active today. We also have fungi, where we can also identify yeast present in our intestines. But we also have a very specific bacteria called archaebacteria, and also protozoa and other more complex organisms. So if we think about our intestines like that, we are talking about a real ecosystem fully populated with these microorganisms. And why I'm saying that? Because when we are born, we are sterile, and we get in contact with these microorganisms after birth. And this perinatal period during gestation, after birth, at the moment of birth and after birth, and I would say for the first 1,000 days of life, is crucial. It's extremely important to establish this ecosystem of microbes and bacteria in our bodies. And this is extremely important and has been studied for many years already. We know that the type of delivery is extremely determinant. It's not the same being born by C-section or being born by vaginal delivery. The primal colonizers of the intestinal gut in the babies are different, and the consequences of that are also very different. So in this period, we have literally a window of opportunity for the modulation of the microbiome, but also for the education of our immune system, which is under development. And then taking probiotics at that time, having a specific diet, taking antibiotics for different reasons, submit the infants different type of stress. All these activities, all these factors can have strong consequences in the colonization of the microbiomes in the baby. So the immune maturation and the microbiota establishment is extremely important in this period of time. The ecosystem I was mentioning is called the microbiota or the microbiome, and I'm going to use these terms a little bit during this presentation. So in the recent years, there was a real focus and attention at the research level on what's going on with this microbiota, what's going on with these microbes which are living with us and which are helping us to function as humans. We have to remember that this microbiota is helping us to digest food. We don't know how to digest fibers if we haven't been colonized by these bacteria. These microorganisms are helping us also to detoxify some molecules. They also have an extremely important role in the education of the immune system because they are basically teaching immune cells. Most of the 70% of 80% of our immune cells are present in the gut. So these immune cells, they need to have this role of sentinels. They need to know what's going on with these microbes in order to understand which are the good ones and which are the bad ones. And scientists, and especially Martin Blaser, professor in New York, have established observation on a chronic loss of the diversity of this microbiota in the recent years. And that was correlated in one side with the increased use and bad use of antibiotics, the level of stress that we have in our societies, the lack of vertical transmission, the increase of C-section birth rather than vaginal deliveries, the increase of different medicines that are used or overused. I mentioned the antibiotics. So all these elements chronically are affecting the diversity of these microbes, and as a consequence of that, this teaching of the immune system is affected. So today, scientists are studying this chronic loss of microbiome, and what we're going to discuss today is how probiotics can help us to reintroduce diversity, either directly by reintroducing lactobacilli and other species, or indirectly by producing molecules that can help other bacteria to be to multiply in our intestines. I was mentioning some of these factors that help us to develop what we call this disbalance of the microbiota in the Western world, the dysbiosis, use of antibiotics, increased eating of ultra-processed food, the obsessive hygiene, something that we need to consider, especially at that time, the stress and the C-section, etc. So today we know that all these factors are decreasing the microbial diversity, and that is on one side. But on the other side, what is interesting is that this chronic loss of microbes has been associated with modern diseases. What we call modern diseases is much more the inflammatory diseases. We're talking about pre-diabetes, type 2 diabetes, obesity, metabolic syndrome, hay fever, inflammatory problems related to the cardiometabolic diseases, neurological disorders. All these diseases have increased in the recent years, and every time we look at the ecosystems, the microbial ecosystems of these people suffering from these diseases, they look much less diverse than the people who are not sick. So today, from the epidemiological point of view, we know that 1 human in 4 by 2025, that means in 4 years' time, will suffer from a non-communicable disease or a chronic disease. And this is based on the inflammatory processes, is based on different etiologies and different causes that are provoking these diseases. And today, we strongly believe here in BioGaia that we need to prevent these diseases. And the question is, how could probiotics work? What are probiotics? So probiotics have been defined by Ilya Mechnikov in the beginning of the 20th century as live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. But today, the probiotics definition has been extended to other concepts like the symbiotics, combining probiotics with fibers, postbiotic, having fragments of these probiotics that could have also a beneficial effect, and combining that with prebiotics, as you know, the fibers that I was mentioning before. So we're going to talk today about the probiotics and how could probiotics help us in this chronic loss of diversity. Last slide of this brief introduction on these concepts is the fact that these microbiomes, these different microbial ecosystems are interconnected. And we know that in each ecosystem, in each body site, we have a specific microbiota. But we also know that we share some microorganisms in one area of the body compared to another area. And today we're going to focus on the interactions between the mouth and the gut, because first of all, because of the oral cavity is the first part of the digestive tract, and second of all, because we're going to mention how probiotics could help in the oral cavity and what could be the consequences of helping us in some of our oral diseases in terms of systemic diseases and intestinal diseases. So to do so, and to dig a little bit more into the data that we have on our probiotics, I have the pleasure to leave the floor to Jacob. Jacob, please.
Thank you so much, Gianfranco, for this inspiring talk. I will restrict myself and go more into detail on the use of oral probiotics in oral health, and more specifically using Leishmania lactobacillus reuteri as a therapeutic in oral health. I will be discussing various properties of these probiotics. I will discuss the immunomodulatory properties, the ecological impact they can have, as well as colonization and adherence to mucosal surfaces. Then Gianfranco will come back to us and discuss a little bit more about future developments and the impact on systemic health. The two most well-studied probiotics for oral health are two Lactobacillus reuteri strains, the DSM 17938 and the ATCC PTA 5289. And how this came to be, that they were so well studied in this particular segment, actually dates back to Japan in the year around 2000. At that time, the dentistry community had a fear of yogurts inducing caries, since they contain lactobacilli. In order to investigate this, Professor Nikawa at Hiroshima University looked at the growth or the effects of growth of various yogurts on Streptococcus mutans. And Streptococcus mutans is one of the microbial drivers of caries. What he found was that most of the yogurts had no impact on the growth of Streptococcus mutans. Some actually increased the growth as hypothesized, and only 2 reduced the growth. And so what were the properties of these 2 yogurts? The common denominator of these 2 yogurts were that they contained the same strain of Limosa lactobacillus reuteri. And that strain was the ATCC 55730. And that is the mother strain of what is now the DSM 17938 that I mentioned in my previous slide. So I will start to discuss the immunomodulation of these strains. As you see here to the left, there are many lactobacilli that provides substrate for the production of short-chain fatty acids in the lumen of the gut. And this, in turn, can affect the induction of regulatory T cells, which creates a suppressive environment of the immune system locally in the gut. Lactobacillus reuteri, and especially the strain, the DSM 7938, can also induce regulatory T cells, but in another manner, and that is through the production of adenosine. And that, in turn, is capable of reducing regulatory T cells in a TLR2 dependent manner. The third pathway, the specific pathway that I would like to discuss, is the pathway of histamine, which at first may sound counterintuitive, since histamine is you would expect that to have a pro-inflammatory response in connection to, for example, allergies and that is true. So, but at the same time as Lactobacillus reuteri, in this case, another strain called 6475. Produces histamine, it also produces an inhibitor that inhibits the pro-inflammatory pathways of histamine, leaving only anti-inflammatory pathways, which causes a net anti-inflammatory response. So how does this translate into the biological situation? Lactobacillus reuteri PTA5289 is potently anti-inflammatory, and as you can see in the graph to the left, it in a biofilm it reduces TNF alpha potently. Not only does it reduce the acute inflammation, it also is capable of reducing the downstream effects of inflammation, as you see in the graph to the right. And local inflammation of the jaw can induce gingivitis and bleeding of the gums and periodontitis, which is tooth loss. And this is driven by inflammation. And as you can see in the graph here, that in the control situation you have a healthy bone tissue, and when you induce periodontitis, in this case in rats, the bone degenerates. And by adding in Lactobacillus reuteri treatment, this can partially be reversed. In the clinical situation, Lactobacillus reuteri can be used to counteract inflammation as well. Also, this has been backed up in the clinical situation in several trials. And this trial that I'm showing you right now is performed by Professor Schlagenhauf in Würzburg, Germany, where he showed in a very, very controlled manner how Lactobacillus reuteri can be used to prevent gingivitis. In this trial, as you can see here, Dr. Schlagenhauf recruited sailors at a ship deployed at sea. And during this time, they were all eating the same food and having very, very, very similar habits, making this an ideal population to study. As you can see in the graph to the right, the bleeding gums, the gingivitis, is dramatically reduced after 14 days of using Lactobacillus reuteri, as compared to the control group where the gingivitis is unaffected. The other aspect of Lactobacillus reuteri that is important to address is the impact on the microbial community and oral pathogens. As you can see here in the graph to the left, the ATC 52730, which is the mother strain of the DSM 7938, is a potent producer of Reuterin, which is a broad-acting antimicrobial substance. To the right, you can see an in vitro experiment where inhibition of Prevotella is potently inhibited by this strain. In the clinical situation, there are several important oral pathogens that are interesting to investigate. And in this slide, you can see data from an Indian trial where these pathogens were quantified. And you can see before and after the use of Lactobacillus reuteri, you have a dramatic reduction of, for example, Porphyromonas gingivalis. So these are all certain species of oral pathogens, but does that mean that we have a general antimicrobial effect, and would that in case be a good or bad thing? This trial that you see here investigated the microbial community more broadly, and what you can see here is that the data shows that on a relative abundance level of various, on a phylum level of various microbial populations, these are unaffected, while again, when looking at certain species and pathogens, as you can see here labeled in red, they are specifically affected. As I started to mention in early on in the Japanese early trials, Streptococcus mutans is an important driver of caries. And this has also been investigated. The use of Lactobacillus reuteri has also been investigated in this situation. As you can see here, after a month of using Lactobacillus reuteri, the counts of Streptococcus mutans is dramatically reduced, almost to half. The final property that I would like to bring up during this presentation is the adherence and the colonization of the probiotics in the oral cavity. And as you can see here, both strains that I've been talking about throughout this presentation are able to adhere to oral mutants. And this is especially true for the strain to the far right that was initially isolated from a patient with very good oral health. Finally, does these probiotic actually colonize the oral cavity? And the answer is partially. They, as you see here in this slide, you can see that during the intervention period, the counts of Lactobacillus reuteri increase, and then they gradually go back once you stop using the product. With that, I will conclude my talk and give the word back to Gianfranco.
Thank you, Jacob. That was very important to put into perspective. First of all, the specificity of probiotic strains. This is extremely important. But also all the data that we are providing with these probiotic strains, not only at the clinical efficacy, but also at the mechanistic level. How does the probiotic strain work? How do they work in terms of this ecosystem? So we are talking about this oral cavity, and I was mentioning at the beginning of the presentation the co-evolution of these microbes with us, with our bodies and with our different body sites. So the oral cavity is one of these examples, and Lactobacillus reuteri strains that co-evolve in the oral cavity, like ATCC PTA 5289, are extremely important because they know what to do. They know how to act and interact with this complex ecosystem of other bacteria. And as you could see in Jacob's presentation, some of them are not good bacteria for us. They could be pathogens, and they could induce diseases, oral diseases like gingivitis, periodontitis, caries, etc. So the question now is how we move forward into that, because we have clinical data, clinically proven efficacy in gingivitis, in periodontitis, etc. So the question now is, okay, we mentioned at the beginning that we are suffering from this modern lifestyle, a chronic loss of the diversity of these microbes, and probiotics could help us to prevent this loss of diversity. First of all, by reintroducing these lactobacilli that sometimes could be lost, but also by provoking and inducing interactions of these probiotics with the microbiota, which is already living with us. And one of the questions we have is, we have been studying different diseases, and in the research community, there is a lot of attention on what's going on with the microbial structure and composition of these microbiotas, especially in the oral cavity, and what is the correlation of losing or gaining some bacteria with some diseases. So the case of periodontitis and caries is extremely clear. Streptococcus mutans, it's increase in the presence of caries. So how lactobacilli can act against Streptococcus mutans? So you already saw Jacob's presentation on that. But the question is, what about other diseases? And what about the potential link and interconnection between our oral cavity, our oral microbiota, with our intestinal microbiota, which is the more described and known microbiota. So today we know that some of these changes in the oral cavity are linked to other diseases. For instance, type 2 diabetes, and I was mentioning the importance of the increase, epidemiologically speaking, of the type 2 diabetes in the world. But we are also talking about, for instance, colorectal cancer, where people who are enriched with some specific pathogenic bacteria like Fusobacterium nucleatum in the oral cavity have been observed that there is a strong correlation, or at least a positive signal, for the colorectal cancer. So with all these, we are trying to link and make connections between, in one side, avoiding this chronic loss of microbiota, and on the other side, bringing probiotics that can help us. And the next steps are really clear for us. We have all this science, which is extremely rich, extremely detailed, and extremely, I would say, technologically driven in terms of understanding how our probiotics are functioning. And that could be related to what it was started as the microbiome revolution in the early 2010, where with this paper in Nature. But what we're talking here is, we're talking about improving people's life. And then that means we need to translate what is published in scientific journals into something which is useful for people. And that's what we are doing in BioGaia, in terms of translating all this science that Jacob was presenting to you into something which could be concentrated in a product and then could be given to people in order to prevent, for instance, some of the diseases related to the loss of this microbial diversity. And that's a kind of engine for us in terms of innovation, and it's extremely important that this translational challenge, or this translational transduction or traduction of what we are doing in science and then making it happen in real life is what we are developing here in BioGaia. So with this, I'm going to stop here. I'm going to thank you for your attention, and I will leave the floor to Caroline.
Yeah, and thank you very much both to Gianfranco and to Jacob for these exciting presentations. And by that, we would like to hand over to Christina, that will present BioGaia and the foundation that prevent antibiotic resistance. So the screen is yours.
Thank you so much, Caroline. So as you understand from listening to my colleagues, Gianfranco and Jacob, BioGaia is a scientifically driven company, and we have developed clinically proven probiotic products for the last 30 years. My interest in this area was actually ignited 20 years ago when my grandmother was diagnosed with colon cancer. I had studied nutrition and microbiology and was certain that there had to be a connection between the diet, our microbes, and the intestines and our overall health. So at that point in time, as Gianfranco also mentioned, the first definition of probiotic bacteria was set by WHO, which defined probiotics as live microorganisms with documented health benefits. And now this area, as you just heard, is exploding with new research, and we are trying to understand the different connections. What we also know from all this research is that all bacteria are not the same, and that different strains has different health benefits. And in order to be named a probiotic, the effect should be proven in clinical trials. However, currently in Europe, no probiotic claims are allowed on food supplements. But with my natural interest in this area and in the microbiota, I'm really happy to be working in a company with a mission to be the groundbreaking leader in probiotics that contributes to improved health by educating people and providing probiotic products for defined indications. And just to give you a brief understanding of what BioGaia is, we were founded in 1990, and today we are around 160 employees. We are listed on the Nasdaq index. And we have more than 500 patents on different bacterial families. We have products in more than 100 markets and offices in Sweden, Singapore, USA, Japan, and South Africa. And we have 3 subsidiaries, BioGaia Production, BioGaia Pharma, and Metabogen, which I will come back to. So I also just want to mention that we have this network business model. So in this map, you see all the yellow spots where we have offices, and the green ones are the research network that we are working with. So we are working with universities and hospitals all over the world, and they are doing research with our strains, and both by themselves and in combination with us. And then the blue spots are where we have production or packaging facilities. And the orange spots are the network of partners where we are working with for distribution and sales of our products around the world. So all the BioGaia probiotics are clinically proven, and today we have more than 200 clinical studies that have been completed, and results have been published in scientific journals. And that also means that in more than 18,000 individuals of all ages have eaten and gotten our probiotic products. So looking at sustainability, BioGaia has since the very beginning seen the emerging problem with antibiotic resistance and the detrimental effects on society. And you could say that it is part of our DNA and our core values. And just to refresh your memories on the situation regarding antimicrobial resistance, WHO states that antimicrobial resistance is a global health and development threat that requires urgent multisectoral action. And WHO has declared that antimicrobial resistance is one of the top 10 global public health threats facing humanity. And the main problem is misuse and overuse of antimicrobials, which are the main drivers. So as a result of the BioGaia commitment to work with this problem, BioGaia established the PAR Foundation in 2017 with the aim of contributing to the reduction of antibiotic resistance. And we are each year donating money to the PAR Foundation that then donate it for research. However, our commitment in BioGaia didn't stop by founding the foundation, so we are still wanting to contribute to solutions based on science. But to give you some examples of what the PAR Foundation are working with now, they recently gave out 3 grants, and 1 grant was given to a project in Ghana on the prevention of pneumonia and this is it will be an exploratory study. Another study is a Swedish project where they will look into prevention of respiratory tract infections. And finally, there's a project in Portugal where they will look into prevention of urinary tract infections in the elderly care. So we're really glad and happy and proud to be part of sponsoring the PAR Foundation. But looking into the future, BioGaia is a group, and we would say that we will be more of a microbiome partner. And in Sweden, we have BioGaia production, which is based in Eslöv, where we can produce our drops, we can produce a mini pack, and we can produce samples. The site is GMP certified. And this is also the place where we have our Center of Excellence. So this is where Discovery and Research and Product Development do their new innovations. They can test and they can make small-scale production. And as a final thing, which I also mentioned in the beginning, we have the subsidiary of Metabogen, which will give us the opportunity also to be part of developing next generation probiotics. Because with Metabogen, we can develop products from novel anaerobic strains derived from human gut and not previously used as probiotics. We can also evaluate potential probiotic strains by metagenomic technologies and metabolic modeling. And we have the opportunity to use unique technology for production of products with these anaerobic strains. And finally, we can do strain developments and small-scale production. So this is a really interesting area for the future. And some of the focus areas for Metabogen are metabolic diseases, microbiome restoration, and intrahepatic cholestasis of pregnancy. So with that, I will finish the short information about what BioGaia is today and what we are also looking at for the future.
Thank you very much, Christina. And I would also like to thank all three speakers. And it's now time for the question and answers sessions.